Yeah, I see it, Jan. Okay, I think we can get started. Let's go on to that. Hello, everyone. Welcome to episode 3, season 1 of Cell Migration Seminars. Same information as always at the beginning I'll give. So just a reminder, if you're watching on Zoom, then you can either ask a question by writing in the chat, or you can write the word question or Q to ask the question yourself. I'm going to very strongly encourage everyone, if they have a working microphone, to please ask a question themselves. It will make it much better and more interactive. So please do that if you'd like. On YouTube, that doesn't have that function, so please just write in the chat. Our contact information is there. And we also have a Slack channel, which hopefully everyone received in the emails for post-seminar discussion. We had lots of it last week after Robert Insel's talk. And if we get the same volume of questions, which I fully expect, then that could be a good platform for discussion afterwards. That's all I have to say. So to introduce today's speaker is Sally Horne-Badovinac. hopefully I made a good attempt at that. Sally is an associate. Sally is associate professor of molecular genetics and cell biology at the University of Chicago. Her lab uses genetic, cellular, and quantitative live imaging approaches in Drosophila to probe the dynamics of epithelial tissues and their contributions to organ shape. And today she'll be talking about the signaling mechanisms that coordinate cell movements for epithelial migration. So I'll stop the share.
Okay.
If you'd like to go ahead.
Why is this not? Okay. All right. So I really want to start out by thanking Adam, Becky, and the whole crew for basically initiating this seminar series and hosting these seminars. I really think that this has been a terrific way to kind of keep the community engaged during this time of social distancing. And I'm really excited to be a part of it. So thank you so much for inviting me to share my lab's work. So as Adam mentioned, I have really a long-standing interest in understanding how organs are formed and shaped during embryonic development. And we have a particular focus on the role that epithelial tissues play in this process. So over about the last 12 years or so, my lab has really been focused on trying to understand how it is that epithelial cells migrate collectively. This is a process that happens in multiple instances in vivo. So during development, you're going to use a motion like this to shape organs like the kidney, the eye, the mammary gland. We know that during adult homeostasis, you're using a migration of this type to drive turnover of the epithelium of the gut, to heal wounds, and that ultimately this type of collective behavior assists in the spread of epithelial cancers. And the aspect of this process that my lab is really interested in is to try and understand how each cell in this tissue signals to influence the behavior of its neighbors. And we think about this happening both at the global level, in terms of signals that act at long range across the tissue, as well as local signals that are acting at short range right between neighboring cells. Now, just to be sure that we're all on the same page, I just want to remind everyone that epithelia are tightly adherent sheets of cells that are polarized along two axes. So all epithelia have a primary apical basal axis with a basement membrane extracellular matrix here at the basal side. And when we're thinking about a migrating epithelium, a lot of the action in the system is going to happen right at that cell matrix interface. So now if we turn the tissue over so that we're focused on the basal side, I can tell you that when an epithelium is migrating, it's also polarized across the tissue plane so that each cell has a distinct leading edge versus trailing edge in the direction of tissue movement. Now, when epithelial cells are migrating, they're using the same basic machinery that we associate with an individual cell in culture. Right? They're making actin-based protrusions at their leading edge. They're using integrin-based adhesions and actin-myosin contractility to generate traction forces against the substrate. But the thing it is that sets the epithelial cells apart is that they need to take all of these individual cell migratory behaviors and organize them across the tissue so that the tissue as a whole can move in a directed way. Now, this can take the form at the global level of basically just getting the front-rear axes of all the cells pointed in the same direction. But at the local level, you also need to have really tight coordination between the leading edge of one cell and the trailing edge of the cell in front. Because when an epithelial cell forms a leading edge protrusion, that structure is actually going to go underneath the trailing edge of that leading cell. Right? So that you get an arrangement like overlapping shingles on a roof. So this makes this region, this leading trailing cell-cell interface, a really particularly important place where signaling needs to occur. Now, the system that my lab uses is the Drosophila egg chamber. This is an assembly of just about 850 cells that's going to produce one fly egg. This structure has an inner cluster of germ cells, an outer somatic epithelium called the follicle cells, And then the follicle cells produce a basement membrane extracellular matrix that acts as a sheath around this entire little structure. Now, when the egg chambers are in the ovaries of an adult female fly, you see that they're arranged in developmental arrays with younger egg chambers at one end and older egg chambers at the other. And what I want you to notice is that as these structures are growing and developing, they're also elongating along one axis. And the reason that they're doing that is to ultimately create the elliptical shape of the fly egg, which is going to set the shape of the embryo that develops inside. Now, the migration we study is going to be of these epithelial follicle cells. And the reason that I'm telling you about this elongation is that that's what the migration is required for. It's why the follicle cells migrate. But I'm not going to discuss this morphogenesis anymore during this talk. We're going to focus on how the follicle cells migrate. And if you'd like to discuss the why at the end, I'm happy to do that during the questions. Okay, so what I'm showing you now, this is a gorgeous movie that was taken by Sayori Hygo and David Builder's lab. And what you're looking at, this is a string of these developing egg chambers in culture. The red balls are the nuclei of those internal germ cells, and the epithelial cells are outlined in green. And what I want you to notice is that these two egg chambers in the foreground are rotating. Now, the reason that the egg chambers are rotating, I'm going to show you, we can take an egg chamber, we can cut it in half, flip it 90 degrees so that we're looking down the long axis. The reason that the egg chamber rotates is because the basal sides of those follicular epithelial cells are motile, and they're crawling on the inside of that basement membrane matrix, which is remaining stationary throughout this process. And so now, if we take one of these rotating egg chambers and we compress it against a slide, we image the surface right underneath the cover slip. That's how we generate movies like this very first one that I showed you in the talk. And once you can look at the migration from this perspective, it becomes very clear that these cells are crawling along that basement membrane in a collective migration to generate that rotational motion. And it's these basal views of this tissue that I'm going to show you throughout the rest of the talk. Okay, so today we're going to talk about two aspects of this collective migration. The first thing that I'm going to do is I'm going to tell you about a planar signaling system that helps to organize all of these cells in the same direction for directed tissue movement. And this is going to be about 2/3 of the data that I show. And then in the second part of the talk, we're going to focus in on what's happening at the level of a single cell and talk about how stress fibers are organized within these migrating epithelial cells. But before we get into the data, I want to introduce you to the people who did the work. So we're going to be talking about work that's been done over a long period of time in my lab by 3 former lab members, Maureen Cetera, Carrie Barlan, and Claire Stedden, as well as newer data from 2 current lab members, Audrey Williams and Kristen Sherrard. Okay, so let's start out with this first topic. So one of the biggest questions in any collective migration system is how you get all of the cells polarized in the same direction for collective movement. And I'm going to argue to you that this is a particularly hard question in the egg chamber. Because when we're talking about these follicle cells, this epithelium is forming a topologically closed tissue around the germ cells. And what that means is that there are no external cues for polarization in this tissue. There's no empty space for the cells to migrate into, no leader cells versus follower cells, no chemotactic cues. More than that, the direction of migration is not fixed. Each egg chamber can migrate in either a clockwise or a clockwise direction. Now, once a direction is chosen, that epithelium is going to continue to migrate in that direction for 2 full days in vivo. But the decision of which way the epithelium is going to go is made on a stochastic basis from one egg chamber to the next. So what we think we're really dealing with here is a self-organizing system where to initiate migration, all the cells in the tissue have to make a collective decision about which way to go. Now, we don't know how symmetry is broken in this tissue to allow that collective decision to be made. But one thing that we do know is that this requires the function of a protein called FAT2. Now, FAT2 is a giant cadherin that localizes very specifically to the trailing edge of each one of these migrating follicle cells. Now, in this location, this protein, although it is a cadherin, it is not functioning as a cell-cell adhesion protein, but rather as a signaling protein that's helping to coordinate the motility of these cells across the tissue. And even though I'm going to be focusing on the work of FAT2 from my lab, I'd like to quickly point out that this protein has been studied in the follicle cells by a number of different labs over many years. But in the context of this talk, what I'm going to tell you about is that FAT2 sits at the trailing edge of the cell and it induces the formation of leading edge protrusions in the cell behind. And in doing this, it's forming the core of a larger signaling complex. So in that way, FAT2 promotes epithelial migration. But then I'm going to show you that epithelial migration also promotes FAT2 signaling by localizing this protein to the trailing edge of the cell. And then at the end, I'd like to speculate how this resulting positive feedback loop may actually be giving us insight into how you initially polarize this tissue for directed movement. Okay, so let's talk about FAT2 and protrusions. So what I'm showing you here is the basal surface of the follicular epithelium stained for F-actin with a single cell highlighted. And what I want you to notice is that each cell in the tissue extends an actin-based protrusion. Now, this happens pretty much the way that you would expect. The protrusion depends on the formation of a local branched actin network under the control of the scar wave complex, which includes the proteins SCAR and ABI. And we know that the follicle cells use this machinery because if we look at the basal surface of the tissue, we can see SCAR very nicely localized to the leading edge of each cell. And even more, if we get rid of the function of the scar wave complex, in this case, expressing an ABI RNAi transgene in a clone of cells shown in green, we can very nicely eliminate these protrusive structures. So just to orient you to how to look at this tissue, this is a wild type cell with a bright white leading edge protrusion right next to an ABI RNAi cell that lacks these structures. Okay. And if we use that same RNAi transgene, we express it in the whole epithelium, we get rid of all the protrusions from all the cells, we now block the ability of the tissue to migrate. Okay, so now what I'm showing you, this is a mosaic epithelium that's a mixture of wild type cells and FAT2 mutant cells. So the wild type cells are here on the left in blue. You can see that they have bright white leading edge protrusions, and the FAT2 mutant cells lack these protrusive structures. And this phenotype is just as strong as what I showed you for loss of the scar wave complex. Now, this can be a little bit confusing because I've told you that FAT2 localizes to the trailing edge of each cell, but the phenotype I'm showing you is at the leading edge of the cell. And the reason that this occurs is because this phenotype is non-cell autonomous. Now you can see this really easily if we look at a clone with a different shape. And first, I want you to focus on this row of wild type cells that's migrating right behind a row of FAT2 mutant cells. Because when we have this arrangement, the wild type cells lack leading edge protrusions. By contrast, if you have FAT2 mutant cells migrating behind wild type cells, the mutant cells can make protrusions just fine. So what these data are telling us is that FAT2 sits at the trailing edge of the cell and it reaches out and it tells the cell behind it where to put a leading edge protrusion. Now, we're obviously interested in how FAT2 performs this function. And so far, I've been showing you FAT2 as a broad chevron at the back of the cell. And in fact, that's not really what FAT2 looks like. If we look at endogenously tagged FAT2 within the tissue, what we see is that FAT2 actually forms these discrete clusters within that trailing plasma membrane. And we were really curious how this clustered organization could induce cell protrusive activity. Now, one possibility is that you take the signals coming from all of these clusters, integrate them across the leading edge of the cell behind to produce a broad protrusive domain. Alternatively, each one of these clusters could induce a local focus of cell protrusive activity. And in fact, I'm going to show you that that's what we think is happening here. So in this image, I'm showing you the overlap between those FAT2 clusters with the ABI component of the scar wave complex. Now, ABI is also punctate within the plasma membrane. This localization is not as crisp as what I'm showing you for FAT2. But if we take line scans along those interfaces, we can see that there's really a very nice correlation between the signals, and we get a correlation coefficient around 0.7. But this localization between FAT2 and ABI is actually even easier to see live. So I'm going to start out drawing your attention to 3 of these green FAT2 spots that are next to the pink ABI spots. And I'm going to just tell you that the signals in this movie have been slightly offset vertically so that you can better see the correspondence between them. And now as the movie plays, I think it becomes really quite clear that each little green spot of FAT2 is being chased down the screen by a little pink spot of ABI. So what we conclude from these data is that this is really a very local recruitment event. Each one of these spots of FAT2 that's at the trailing edge of one cell is locally recruiting a focus of the scar wave complex, and this is leading to a local focus of cell protrusive activity. Now, there's obviously something missing from this picture because I'm telling you about a transmembrane protein in one cell recruiting a cytoplasmic complex in the cell behind. So clearly there need to be some sort of transmembrane receptors at the leading edge of these cells that are transducing that FAT2 signal. And this is going to take us to the second part of this talk, where we're going to talk about the larger signaling complex to which FAT2 belongs. Now, so far, we've identified 2 other transmembrane signaling proteins that are required for the migration of the follicular epithelium. These are a receptor tyrosine phosphatase called LAR and a semaphorin protein called SEMA5C. Now, both of these proteins localize to the leading edge of each migrating cell, and they're also both involved in setting the site of where the protrusions are going to form. Now, the way they do this is a little bit complicated, so I'm not going to focus on this aspect of the signaling today. The thing that I want to show you, however, is that FAT2 is actually responsible for recruiting both of these proteins to their leading edge location. And because the data look exactly the same for both LAR and SEMA5C, I'm only going to show you what this looks like for LAR. Okay, so just like FAT2, LAR forms nice discrete puncta within the plasma membrane, and these puncta beautifully overlap with those of FAT2. And again, if we take line scans along these interfaces and look at the overlap between the signals, we're getting a correlation coefficient close to 0.8. So it's really a very nice co-localization. But what's even more convincing is FAT2 is required to stabilize LAR at the basal surface of the tissue. So I'm once again showing you a FAT2 mosaic epithelium where we have wild type cells in blue on the left, FAT2 mutant cells on the right. You'll notice that LAR localizes normally in the wild type cells, but it's largely missing from those FAT2 mutant cells. And once again, this is a non-autonomous phenotype. So if we first focus on these 2 wild type cells that are migrating behind FAT2 mutant cells, we can see that these wild type cells lack LAR at their leading edge. By contrast, if you have FAT2 mutant cells migrating behind wild type cells, the mutant cells can localize LAR just fine. So what we have is a situation where FAT2 sits at the trailing edge of the cell, it reaches out and stabilizes LAR in the leading edge of that cell behind it. Okay, So again, without showing you the data, I'm just going to tell you that the exact same situation is true for SEMA5C. So there's a couple of things to mention about these data. The first is that we don't know that this interaction between FAT2 and the leading edge proteins is direct. There could be 20 other proteins in this complex that we haven't discovered yet. We have no biochemistry at this point. But the other thing that I really need to tell you is that this relationship is not reciprocal. FAT2 is required to localize LAR in SEMA5C. LAR and SEMA5C are not required to localize FAT2. And this really has made us start to believe that FAT2 is the stable core of this signaling complex. And so we wanted to look at this a little more closely by looking at the relative stability of these proteins in a FRAP assay. Okay, so at this point, we have all three of these proteins endogenously tagged with 3 copies of DFP, so we can do direct comparisons between them. And what I'm going to show you is a FRAP experiment where we're going to photobleach the cell-cell interfaces that are highlighted by the yellow boxes on the screen. And as this movie plays, you can see the flash of the photobleach, and then the fact that LAR is returning to the tissue relatively quickly. And if we look at these data in graphical form, what we can see is that LAR and SEMA5C have very similar recovery dynamics, and they're coming to a stable level of recovery fairly fast, just about 3 minutes after photobleaching. But in this first experiment, by the end of 8 minutes, FAT2 still hasn't even come back to a stable level. So if we take this experiment out longer and we only look at FAT2, we can see that FAT2 eventually comes to a stable level of recovery about 12 minutes after photobleaching. But even once it's stabilized, it's not recovering nearly to the same extent as LAR and SEMA5C. So this really suggests that FAT2 is much more stable. LAR and SEMA5C are coming in and out of the complex much more rapidly. Okay, so at this point, we're really pretty convinced that FAT2 is the dominant player, at least among the proteins we have found so far. And that's really led us to believe that the localization of FAT2 to the trailing edge may be a really key component of this entire system, because it's by localizing FAT2 that you recruit these transmembrane proteins and the scar wave complex to kick off setting up the protrusions for the entire tissue. And so that made us really interested in understanding how FAT2 becomes localized. And so far, the major thing we've found is that the migration of the tissue itself is required to localize FAT2. Okay, now the way that we know this comes from looking at 2 genetic conditions that I've already shown you in this talk. In the first condition, we are going to remove the scar wave complex from all the cells, and that's indicated by the yellow dots on the cells. This eliminates the leading edge protrusions from the entire tissue, and it blocks the ability of the tissue to migrate. And we're going to compare this to a condition where we are only removing the scar wave complex in a small clone of cells. Because in this case, you remove the protrusions from those cells, but the protrusions on all the rest of the cells allow the tissue to continue to migrate, and you just carry those non-protrusive cells along the migration path. They're wild type neighbors, or basically just carrying them. Okay, so what that means is if you see a phenotype in the first condition that you do not see in that mutant clone, you know that that phenotype is caused by the loss of tissue motility, not by the loss of the scar wave complex. Okay, so what I'm showing you now is an epithelium where we've removed the function of the scar wave complex in the entire tissue. And you'll notice that you lose that planar polarization of FAT2 across the tissue. Now, on this scenario, FAT2 still organizes into those discrete clusters. The discrete clusters still recruit LAR. The only thing that is defective here is the polarization of those complexes across the tissue. Now, in this second condition, what I'm showing you is we've removed the function of the scar wave complex in a clone of cells, in this case through a scar mutant allele, And now FAT2 is still polarized normally to the trailing edge. So this tells us that FAT2 does not require the scar wave complex to localize if the tissue is moving. Okay, so from these data, we can conclude at the very least that migration of the tissue is required to maintain FAT2's localization to the trailing edge. But what we'd really like to know is it also required to initiate that localization pattern? And typically this would be a fairly easy experiment, right? You just ask, is FAT2 polarized before the tissue starts migrating, or is the tissue migrating before FAT2 becomes polarized? But it turns out that this is actually kind of a hard question to ask within our system, because at the time that the migration initiates, the egg chamber is so small and so round that it's really very difficult to image the basal surface and watch the dynamics that are happening. So to get around this obstacle, my lab took advantage of kind of the fun genetics in Drosophila to temporarily, basically to block migration early in egg chamber development and let it reinitiate at a time when the egg chamber is much more amenable to imaging. So the way that we do this is by expressing a FAT2 RNAi transgene just in the early stages of egg chamber development. Because we have FAT2 endogenously tagged with 3xGFP, we can watch the protein come back to the tissue as that RNAi starts to wear off. And we can watch what's happening with the polarization of FAT2 in the context of migration initiation. And to give you a sense of what this looks like in real tissue, I'm again showing you one of these strings of egg chambers in culture. You'll notice that the youngest two egg chambers have no FAT2 expression. This third egg chamber is starting to have a little bit of FAT2 expression. And then in this fourth oldest egg chamber, FAT2 is back to normal levels and it's polarized to the trailing edge. And if we watch a movie, you can notice that it's only this older egg chamber that has the high FAT2 expression that's rotating. All right, but what's happening in that third egg chamber where the FAT2 is just starting to come back to the tissue? So when we look at egg chambers right at this stage, it's really interesting because we can see that as the epithelium is starting to begin a slow directed migration, at least in some parts of the tissue, FAT2 is not yet robustly polarized to the trailing edge. So we really are starting to have the impression that migration initiates at least at a slow pace before FAT2 is truly localized to the trailing edge. Okay, so I hope at this point I've convinced you that this feedback loop between FAT2 signaling and the migration of the tissue actually exists. And now we're really interested in trying to understand whether this can help us to think about how this tissue becomes polarized. Now, FAT2 induces the formation of a protrusion in its direct neighboring cell. So this is an incredibly local signal. It is one cell talking to its direct neighbor. But you can imagine that if that signal induces motility in that cell, and that motility in turn localizes FAT2 to the trailing edge to begin to polarize the cell behind it, you can see how this local signal could begin to propagate. And then once the entire tissue is moving robustly, this provides a really nice mechanism to keep that migration moving in the same direction over long periods of time. Because as new FAT2 protein is produced and moved to the cell surface, it's brought to the back, and then that helps to keep this persistent motion going in one direction over the course of that 2 full days that this migration needs to happen in vivo. So we're really interested in testing this model going forward and understanding what it is mechanically about migration that is ultimately localizing FAT2 to the trailing edge. Okay, so now what we're going to do is we're going to switch gears and we're going to focus now on how stress fibers are organized within individual cells in the tissue. Okay, So for those of you who've been paying attention, as I've been showing you these phalloidin stains of the basal surface, you may have noticed that in addition to having a leading edge protrusion, each one of these follicle cells also has a beautiful parallel array of stress fibers at its basal surface. And in the same way that these protrusions are all oriented across the tissue, these stress fibers are just in a beautiful array across the tissue and oriented in that direction of tissue movement. Now, as far as we can tell, the stress fibers in the follicle cells are most similar to the ventral stress fibers that are seen in cultured cells. And just as a quick reminder, these are large actin-myosin bundles that are anchored on their ends by integrin-based focal adhesions to the underlying extracellular matrix. And although we know that these structures are present in just gobs of migrating cells, it really is my impression that we have a very poor understanding of how these tightly adherent structures are remodeled as the cell moves forward. And I think that within the egg chamber, we really have a nice system to look at stress fiber remodeling in the context of a migrating cell. And the reason that I say this is that these follicle cells are simple, small, and natural. So unlike a fibroblast, which can turn frequently and move at uneven speeds, each one of these follicle cells is essentially undergoing a 1D migration with a uniform speed. And what that means is it makes the individual stress fibers very easy to track. On top of that, again, these cultured cells tend to have very large migratory surfaces, but that basal surface of a follicle cell is only about 5 micrometers across. And what that means is that these cells are going to migrate 5 cell lengths over the course of an hour, which allows us to watch multiple rounds of stress fiber remodeling in a very short time frame. And then ultimately, this system is natural. These cells are crawling on their native substrate. So when I show you these dynamics of what myosin looks like on the stress fibers, we feel very confident that what we're looking at is a natural process and that it may in fact represent a general paradigm for how stress fibers are remodeled in migrating cells in vivo. Okay, so consistent with these structures being ventral stress fibers, we know that they are anchored to the basement membrane through integrin-based adhesions. But unlike this classic model for ventral stress fibers, where you only have those focal adhesions at the ends of the actin-myosin bundles, in the follicle cells, each one of those fibers has multiple small adhesions along its length. Now, this paxillin signal that I'm showing you is overexpressed. And the reason that we do that is that it makes the adhesions a little bit bigger and easier to see. But we can see this same pattern with endogenous tags on paxillin, talin, beta integrin. So we really do believe that these multiple adhesions are real and that they're ultimately the key to the dynamics that I'm about to show you. Okay, so what I'm showing you now, this is myosin labeling the stress fibers in an individual cell. Now, this cell is still within the epithelium. We've only isolated it optically to make it easier for you to focus on what's happening at the one cell level. It's a little overwhelming to see all the stress fibers at once. And what I want you to notice is that the individual actin-myosin fibers are persisting as the cell migrates down the screen. And they almost just look like they're gliding along with the cell as it moves. Now, we know that that can't be possible, right? I mean, I've just shown you these things are anchored to the basement membrane through integrin-based adhesions. So the only way the dynamics like this could be happening is through a treadmilling type mechanism, where the entire stress fiber is shrinking at the back and growing at the front. And that's really where we think that these multiple adhesions come in. Because if you think about this classic ventral stress fiber model where you have the adhesions at the end, it's really hard to imagine how you could generate these seemingly gliding dynamics. Okay, so the way that we're thinking about this is that these multiple adhesions along the stress fiber basically imbue them with a modular architecture that allows you to selectively remove old adhesions from the back and add new adhesions to the front. And in this way, a stress fiber can persist over long periods and move along with the cell such that both of them move forward efficiently together. Okay, so to show you what this looks like, I'm now showing you a movie where all of the stress fibers in the tissue are labeled in magenta for myosin, and only a clone of cells is giving you that green paxillin signal. And when we have a movie like this, we can identify a single stress fiber based on the myosin signal, and then we can watch the adhesions associated with that stress fiber form and disassemble over time. And so I'm going to show you what this looks like with a series of chymographs. Okay, so what you're looking at now, this is a chymograph of the adhesions associated with a single stress fiber over time. And this diagonal that you see going down the screen represents that motion of the cell moving forward. Okay, if we focus in on a single adhesion, what I'd like you to notice is this adhesion is born near the leading edge of the cell. It stays in place, kind of locked in position as the cell migrates forward over it, and then it begins to disassemble as the trailing edge of the cell approaches. And we can see the same thing happening again and again and again as new adhesions are added to the front of this same existing stress fiber over time. Okay, so now what I'm showing you, this is a chymograph of the myosin associated with a single stress fiber. And once again, we can see this periodic loading of myosin at the front, and then persistence as the cell migrates over. And I can just tell you that each one of these new additions of myosin is interdigitated with the formation of those adhesions that I just showed you on the last chymograph. Okay, so finally, I want to show you one more chymograph. And this time, we're looking at the adhesions associated with one stress fiber. But this time, I want to draw your attention to this very last adhesion at the back of the cell. So these adhesions are typically larger than what we see in the center of the stress fiber. But even more interestingly, these adhesions travel with the back of the cell, sliding along as the cell moves. And in fact, these adhesions are interesting. Not only are they showing this sliding dynamic at the back, but more often than not, they're also born at the back of the cell. So we can see one of these new sliding adhesions forming here. As it comes to the back of the cell, the older sliding adhesion disassembles, the new sliding adhesion takes over, and it too begins to slide. Now, these types of sliding adhesions have been seen in many types of migrating cultured cells. I'm not sure we expected to see it in the epithelial context. But yeah, we're definitely interested in why these adhesions are forming and what they're doing. Okay, so what we know so far is that not only do these stress fibers have a modular architecture where you can add new adhesions at the front and remove old adhesions at the back, but there's actually two forms of adhesions. There are what we're calling front-forming adhesions that remain fixed in place as the cell migrates over them, and back-forming adhesions that slide with the trailing edge. Now, obviously, we're interested in how both of these adhesions form and how they have these discrete dynamics. But at the moment, we're kind of focused on trying to understand how it is that you add a new adhesion to the front of an existing stress fiber. Now, we don't know. Everything that I'm going to tell you at this point is pure speculation. But we imagine a scenario where you have nascent adhesions that are forming right at the leading edge of the cell. And we can imagine that that youngest stress fiber or the youngest adhesion within a given stress fiber could basically reach out and capture the next nascent adhesion and force it to mature into the stress fiber itself. Now, one way that this could happen is, of course, there are reports in the literature that formins can localize to focal adhesions. And so if you had a scenario like this, where the formin was producing actin filaments that would naturally form an anti-parallel array between those two adhesions, the addition of myosin could then bundle those actin filaments, cause the nascent adhesion to mature, and entrain it into an existing stress fiber. Now, as I said, at this point, this is pure speculation, but we have started to test this idea just by looking for a formin that's required for the formation of the stress fibers in this tissue. And so far, we have found that the formin BAM seems to be at least part of this process. So what I'm showing you here, this is a mosaic epithelium where we have wild type cells in blue on the left and BAM mutant cells on the right. And you can see that the F-actin within these stress fibers is strongly reduced compared to the wild type cells. We can overexpress an activated form of BAM, again in a clone, and we see massive overincorporation of actin into the stress fibers. In this case, I'm not even showing you the clone boundary because the phenotype is so obvious. So at this point, we think we're starting to get a handle on some of the assembly machinery that's involved in this process. And now we want to start integrating it into this modular, formation of the stress fibers and understanding how the whole system comes together. Okay, so just to summarize what I've told you today, I've told you about two different systems that we think are both involved in organizing the cytoskeletal machinery for collective migration. The first case I told you about was a planar signaling system involving a giant cadherin FAT2 that sets the sites where protrusions will form in the tissue and functions in a feedback loop with cell motion to ultimately polarize all those tissues in the same direction across the tissue plane. This second mechanism is happening on the single cell level, where we have a treadmilling of stress fibers to allow those individual stress fibers to persist over long periods of time as the cell migrates forward. And we believe that this, in many ways, that once you have all those cells organized across the tissue, can then help to enforce that cell to maintain a linear trajectory and once again feed into the collective motion of the tissue as a whole. And so we're really interested over time in trying to learn more about both of these mechanisms and how they're integrated ultimately to allow the epithelium to move in a very directed way within the body. Okay. And so with that, I'd like to thank everyone in my lab and particularly highlighting the 5 researchers who contributed to the work that I talked about today. Want to thank my funding sources and all of you for taking some time out of your day to sit and listen to this talk. Thanks very much.
Thank you, Sally. That was great. If everyone wants to unmute and applause. Okay, lots of questions to ask. So I'll start on Zoom and Jen can ask them from YouTube. So a question from Robert Insole about scar wave. So is the scar wave complex stable or does it turn over like LAR or faster?
We haven't done FRAP experiments with the scar wave complex. I would suspect that it's turning over very fast, but we don't know. We haven't looked.
We've got a question from Allison Julio asking about what is the phenotype for the embryo when you stop the migration of the epithelium?
Well, it depends on how acute the migratory block is. So in a case like FAT2, where you get absolutely no migration of the tissue, the embryo is spherical, or the egg is spherical, and it never gets to the point in the oviduct where it gets fertilized. So there is no embryonic phenotype in that case. However, you can do partial knockdowns of FAT2 or other mutants that have less severe phenotypes, you just get a shorter egg. That egg can be fertilized and you end up with a rounder embryo. For the most part, the embryo is fine. You can see some minor effects. So there's been work done by Adam Martin's lab looking at gastrulation movements in that case, and there are some minor effects on gastrulation. There's also been work from Tim Saunders' lab looking at the AP patterning of the embryo. You do see some minor defects just from changing that aspect ratio of the egg. But ultimately, larvae hatch and they grow up into flies. So they don't seem to care that much.
Question from Max B asking about how much rearrangement there is within the cell layer. So do cells rearrange themselves or do they stay static?
Yeah, so very little. There is almost no rearrangement in these cells as they're migrating. Now we know that there is some because the tissue is also elongating. And so we know that the epithelial cells have to be undergoing some rearrangement to get that elongation process to occur. But on the time scales that we're watching the migration, they're really not exchanging neighbors.
Great. Actually, before we move on to the next question, I just wanted to take a second and say what a wonderful, just absolutely stunning talk that was. You're so clear and that was really, really great. So thank you.
Oh, thank you.
All right, another one from, from YouTube. We've got Ankashi asks about the role that stress fibers have in establishing the fat to polarized direction.
Yeah, so we don't think that they do, other than the fact that, you know, if they are contributing to motility in some way. And I'll just tell you, we've never been able to get rid of the stress fibers to such a degree that we can block the migration of the tissue, right? But to the degree that they actually promote migration, because we assume that they do, it would be involved in localizing FAT2 that way, but we don't believe that they play any direct role in localizing FAT2 to the trailing edge.
Okay, I'm going to go to a couple of questions to people who want to ask questions themselves. So Tim Eichen, you have a question?
Yeah, can you guys hear me?
Yeah.
Yes.
So I'm Tim. I'm a graduate student at Hopkins, and that was just a really wonderful, wonderful talk. So I'm wondering about your model for focal adhesion formation at the leading edge. It kind of predicts that you can change the frequency of focal adhesion formation by modulating either, formulin levels or activity. Have you observed that through your experiments at all?
I'm sorry, levels of what? Levels or activity of which?
Of formin.
Oh, formin. Well, definitely, when we express that activated form of formin, we are getting much more stress fibers, but we haven't looked at that live to really see the dynamics of adhesion formation. But that's a good idea. We should probably do that. That might be really interesting.
Right, because if you change the formin levels either up or down, you should expect a higher frequency of focal adhesion formation. Is that correct? More focal adhesion.
Sure. I'm not sure what I would predict just based on the formin levels. But definitely we could empirically look and just find out.
Right. Yeah. Cool.
We've got a question from Tim Fessenden. He says, beautiful results, Sally. Do you see the dam-dependent nascent focal adhesion capture as roughly analogous to that same process that happens for nascent adhesions in mammalian cells?
Yeah, hi, Tim. Yeah, I think that is kind of how we're thinking about it. That's certainly the inspiration for the hypothesis. I'll be honest, we have a really hard time seeing nascent adhesions forming in this tissue. We think that they may just kind of be below our level of detection. So we haven't been able to visualize this happening. When we're really trying to watch that first, adhesion get added, we're kind of seeing the myosin and the adhesion coming up at the same time. And so we're trying to disentangle that, but yeah, we're presuming that it's similar.
From Roberto Mayo.
Hi, Sally. Nice to see
Roberto. Nice to see
talk.
you too.
So I have a comment and a question. I mean, the comment is that you have a very nice system now where you can study how the migration starts. I mean, with this system that you described. And related to that is, and my question is about the shape of the cells. Have you seen when you see the beginning of migration, when the migration starts, have you seen any change in the shape of the cell, or in the early stages where the cells are not migrating? That because when they are migrating, the cells of the shape is very regular.
Right.
So does it change that? I mean, I was thinking in something kind of unjamming effect that could be involved in the beginning of migration, the start of migration.
Yeah, so we haven't looked at that specifically, cell shape at that point. I mean, it's well known that the cells, when they're migratory, do become elongated along their front rear axis, and that they're much more isometric when they're not migratory, or at the very least, the polarization of that, you know, long axis is not within the direction of migration. And yeah, if you're trying to get at, you know, what Jeff mentioned the other day, this idea of the cell aspect ratio and the unjamming transition, you know, in thinking about that, the real question I think becomes, do these cells ever unjam? And I would argue that this is not an unjamming transition. These cells are very jammed. They don't have that liquid-like property. They're not swirling past each other. And that, you know, that this is much more of a flocking behavior. And in fact, in talking with Jeff recently in a meeting, he agreed and said that he really thought that this was a solid flocking behavior as opposed to an unjamming transition. So
yeah, if I can add a
lengthening.
So I was thinking more at the beginning, not when they are migrating, but maybe if what triggered the migration maybe would be an unjamming, and once they start to migrate, they become jammed again.
Oh, that's interesting, that it would be a transient thing.
Yeah, exactly.
Well, certainly we don't see any higher neighbor exchange at the point of migration initiation than we do normally. Yeah. So, but whether or not they lengthen before they move, that's something we could easily look at.
Okay, thank you.
Yeah.
All right, we have a question from Giorgio asking if there are any lateral protrusions playing any role in the collective migration.
Hi, Giorgio. You know, it's interesting because we do occasionally see lateral protrusions, meaning still at the basal surface, but on the sides of the cells instead of the front rear axis. So we don't see these protrusions in fixed tissue with a phalloidin stain, but we're getting much better at monitoring protrusive activity live. And you definitely do see occasional lateral protrusions. We're not sure what they mean. We don't know if they're important or if they're just some byproduct of not localizing that protrusive activity as efficiently as you would like. But definitely, we think that they're part of how you're getting the tissue organized to begin with, right? Because when migration is initiating, protrusions are going sort of every which way. And so in that way, yeah, they're protruding all the way around.
I have a question from Benoit asking about confinement. So does confinement play a role in the symmetry breaking process? And does confinement vary over time at different developmental stages?
Yeah, hi Benoit. I think they have the same level of confinement all the time would be my argument, right? That they're always in this closed epithelium. And so or are you speaking about specifically confinement of the protrusion? Because I know you guys are thinking about that. In terms of just the cells being confined, they always have nowhere to go. They're always surrounded by their neighbors. But whether the protrusions are confined under the trailing edge of the other cell, yeah, I would imagine that that's pretty much always the same as well. Yeah.
We've got questions from multiple different people, all sort of asking a version of the same question, which has to do with if you were to force FAT2 expression, say maybe on the sides of the cells, could you polarize them differently, possibly even controlling or changing the direction of rotation?
Yeah, God, we would love to do that experiment. at this point, we don't have any good ways to move FAT2 around the cell. It's kind of embarrassing to say that this protein is so large that we still don't have like a cDNA where we can go in and build transgenes that would let us move this protein around. We've done other versions of that experiment where if you remove the ICD of the protein, it will start to kind of snake down the lateral membranes a little bit. And then, yes, you do see that it can induce kind of ectopic protrusions on the lateral side a little bit. Yeah, but no, we don't have control. I wish we had control.
A second question from Giorgio asking, are the sliding adhesions dictating the position of FAT2 and in turn of the scar protrusion?
Yeah, so I mean, we've looked and we don't think that the sliding adhesions and those FAT2 clusters, they don't physically overlap that we can tell, right? They seem to be kind of separate structures. But I think we need to do a little bit more mapping to really understand the relationship between those two different events happening at the trailing edge of the cell.
We have a question from Tony Tsai asking about the ECM. What kind of ECM is there? And also, is it oriented in the direction that the cells are moving?
Yeah, okay. So this is a standard basement membrane matrix. You've got your type 4 collagen, laminin, nitogen, perlecan, right? Just like you would find under any epithelial tissue. And yes, it is absolutely polarized in the direction of migration. So as these cells, it doesn't start out polarized, but as the cells are migrating across that epithelium, they do polarize its structure. They're secreting new basement membrane proteins as they're migrating, and those new basement membrane proteins become organized into these linear fibrils on the inside of the basement membrane. And this is actually thought to be, one of the major links between that migration of the tissue and the ultimate elongation of the egg chamber. Now, you would think that because they're polarizing the matrix, that polarized matrix would feed back into the migration of the cells. So far, we don't have any evidence that that's true, which was really surprising, I think, and disappointing to us. But we can get rid of those large fibrils out of the basement membrane. The tissues still migrate at the same speed as it normally would. It doesn't even affect speed or coherence. Yeah, it's a little bit puzzling, but that polarized matrix is ultimately feeding into the elongation of the tissue.
Second question from Benoit asking about whether you see any cases where stress fibers are oriented perpendicular to the direction of motion, like in any mutants.
Yeah, definitely there are mutant conditions that lead to higher levels of disorganization in the stress fibers. And we're really starting to play with those to really try to tease apart the dynamics of how they're forming and disassembling. Trying to think if they're ever truly parallel. Certainly not the whole array is parallel. Basically, you can see that the array just kind of tracks less well with the leading edge is one thing that you can see. You can also see that there's kind of more crossing of the stress fibers, but I don't think that the whole array ever ends up perpendicular to the direction of migration. They're never quite that messed up.
So we have another question from YouTube asking about if you think there's a scenario where it's possible where a part of the epithelium population migrates and another part doesn't.
Yeah, so certainly you can see one part of the epithelium showing a higher degree of movement than the other part. But but pretty much these cells have to all migrate together or not migrate at all. We have some unusual mutant conditions where we can get parts of the epithelium to move at different speeds. So there's a kinase called misshapen that's required to reduce integrin levels at the basal surface. And if you modulate misshapen levels so that you just increase integrins at the basal surface just a little bit, you can get a patch of cells that's migrating slower than the rest of the tissue. And in that case, the junctions will remodel, The rest of the cells will flow past that slow moving clone while that slow moving clone continues to go. So that's one really weird condition where we can force it, but the vast majority of the time the cells all go together or they don't go.
We've got a question from Suvam Mukherjee, sorry if I'm pronouncing that wrong, asking about CDC42, so whether the activation of CDC42 happens in all the polarized cells irrespective of their position.
Yeah, I would say we haven't looked at CDC42 very much. I'm not really sure what CDC42 does in the system.
Excuse me, can I ask that question?
Yeah, go for it.
To Basally? Yeah, so what happens in case of wound healing, when the cells, they get a mechanical void, of the particular GTPase CDC42 is being activated. So my question was, when we see a concerted movement of the cells, irrespective of whether they were in the first line, second line, or third line, does there is a activation of CDC42 in all the cells or only in the first line of cells? That was my question, but you already answered that it wasn't checked. So
Yeah.
okay, so that is what my question was. Thank you.
Yeah, but in this case, you know, we can't really think about like a first row, a second row, a third row, the way that you can in a wound healing scenario, right? Because all the cells are behaving equivalently. So if CDC42 does play a role in the migration, I would expect it to be activated in all cells all the time, because as far as we can tell, there's no real difference between any one cell in the tissue in terms of how it contributes to the motility.
Thanks.
So Abdul says that it looks like there's a gradient in the length of the stress fibers, with the longest stress fibers along the center line of the cells and the stress fibers getting shorter as you go sideways, and wondering if there's a corresponding change in the Paxillin behavior as well.
Well, yeah, I think to a certain extent that's true, that in general, when you look at these epithelial cells, you know, if you think about kind of a classic hexagonal packing, you have the vertex of the cell most often kind of pointed in the direction of migration. And so if you have those two vertices on either side of the hexagon, that is a longer position than what you have down here in these two sides. And so, yeah, I think it's possible that those stress fibers in the middle are a little longer. But I don't think we've noticed the dynamics being particularly different between stress fibers in the center and on the sides, but I would have to ask Kristen Sherrard, who's doing this work, but she's never mentioned it.
I think we can go to the last couple of the questions. So last one from me is from Steve Tavano, asking whether the protein complexes at the trailing edge are generally more stable than those at the leading edge.
Well, in the case of, are we talking about the stress fibers or the signaling?
It doesn't specify, but I'm going to guess the signaling.
Yeah, so definitely FAT2 is much more stable than Lauren Sema 5C. That came out of our FRAP experiments, that FAT2 is much more stable.
Okay, so we have, I just lost it. We're trying to make sure that we got all the questions here.
I think this can be the last one.
Yeah, I think this will be the last one. From Karen Stones, she asks if there's a pattern to the recovery of cell migration relative to the curvature of the egg chamber as the FAT2 RNAi fades away.
Oh. I think not that we've really noticed. I mean, curvature certainly has some effect on the migration of the cells. The cells that are in the center of the egg chamber have a much longer migration path than the ones that are out at the poles, right? And when you're thinking about kind of the total angular motion. So I think that that's always something to consider, is that those cells in the middle are migrating faster than the ones out on the ends, but I don't think we've noticed anything in terms of the recovery.
You could imagine some weird bulging happening though, if you know, if the corset isn't tightening properly in some areas as opposed to others.
Yeah.
Great. Thank you very much, Sally. That was a really fantastic talk.
Thank you so much for inviting me. This was a lot of fun.
Thank you. And thank you for everyone for attending. Next week, we will have a double header. We've got Brian Camley and Anne Revesat giving 2 talks over the course of the hour. So thanks everyone. If you have any further questions or discussion, please head to the Slack channel. Otherwise, we'll see you all next week.
Okay.
Thank you, Sally.